Hello again. In this video clip, we're going to continue talking about viral replication and really kind of emphasizing the diversity that's out there among viruses and how they replicate in their host cells. And we're going to continue the story by going over some example viruses. Now, these are not covered in your textbook. These are some examples I'm pulling out just to home in further on some of the diversity that exists out there about how different kinds of viruses accomplish the steps of their life cycle. And really, I'm pulling in some information here too from the back of the book, some of the later chapters where this information would be described in your text. All right, so the first example I want to talk about are the herpes viruses, and we're going to discuss these as an example of a group of viruses that are double-stranded DNA viruses. So that means their genomes are double-stranded DNA, just like the genomes of their human host cells. And the herpes virus, this is a group of viruses that's pretty large. So yes, it includes the herpes viruses that we're used to thinking about when we hear the word herpes, the ones that cause genital herpes, also your fever blister viruses. If you are a person who gets fever blisters from time to time, that means you have been infected by types of herpes viruses that cause those. Chickenpox is actually a member of this group of viruses as well, sometimes called herpes zoster. So keep that in mind also. This is a pretty diverse group, and then they all carry out their life cycles in a fairly similar way. So I've got kind of the text over here on the right-hand side of the slide. I'm not going to read through that for you, but you can kind of read through that to get an overview of how these herpes viruses replicate. All right, so the first thing, if you're a herpes virus, you've got to attach to your host cell. And remember we talked about some of the different ways that viruses enter their host cells. Herpes viruses are enveloped, so they attach to their host cell via the receptor, and the envelope of the virus fuses or kind of melts into the plasma membrane of the host cell, and then the capsid comes out. So there's the capsid, the protein shell with the DNA on the inside. And interestingly, those capsids slide down the cytoskeleton of the host cell. And if you studied cytoskeletons inside eukaryotic cells in earlier classes, maybe A&P, those are protein-shaped tubes for the most part, or fibers that provide some of the framework and other functions inside the cell. So, viruses are all about hijacking natural features and functions of their hosts to get to carry out the processes that they need to do. So capsid apparently attaches to the cytoskeleton, slides toward the nucleus. The capsid comes inside the nucleus through a nuclear pore. Maybe you've talked about nuclear pores in classes before. Those are fairly large holes that exist in the membrane of the nucleus. All right, and so once the capsid's in here, the DNA will come out. Okay, the viral DNA is out. It's double-stranded DNA. And so you're going to have transcription that's going to take place inside the nucleus. And they're showing RNA polymerase here on the diagram because that's the enzyme that does transcription. It's going to read the herpes virus genes on its DNA genome and transcribe those to make messenger RNAs. And here on the diagram it mentions early and late mRNAs, and what that's telling us is some of the herpes virus genes are expressed early during infection and some later, so that everything is timed properly to complete replication and assemble new virus particles. So those messenger RNAs have to go out here into the cytoplasm, and here we have protein synthesis. So these are supposed to be ribosomes. Remember, viruses don't have their own ribosomes. They're dependent on their host cell's machinery to accomplish that. And then what they're showing you here is you make all these proteins of the virus. Well, those capsid proteins that are made have to go back into the nucleus because your DNA is in here replicating. It's being replicated. And so now you need new DNA genomes to get packaged up inside these new herpes virus capsids. The capsids come out of the nucleus. Now here's something that's unusual. Now we've talked about budding. And when we looked at budding before, budding always took place here at the surface of the cell. So we would expect the capsid to travel up to the cell membrane and push itself through the cell membrane. The cell membrane would wrap itself around the capsid, and that's where it would get its envelope. But herpes viruses do things a little bit differently. They actually bud into the endoplasmic reticulum. So that is a membrane, the ER, the endoplasmic reticulum, is a membrane-covered compartment that's pretty extensive inside the host cell. And it plays a role naturally in the cell as the first part of an assembly line for proteins that the cell makes, things like hormones and so forth, or cell signaling molecules that would need to be secreted to the outside. And so these herpes viruses are kind of hijacking that process as well. They bud into the endoplasmic reticulum, so they push themselves into this membrane-covered compartment, and then they hijack the cell's natural processes that would ultimately send these to the surface of the cell. And they undergo exocytosis, so they get secreted, kind of like a protein signaling molecule or a hormone. If this was a hormone-producing cell, the virus is tricking the cell into thinking that it's just a protein that it's made so that it can work its way out of the cell. All right, and so now here are all these new herpes viruses that have been made. All right. So you can see you're fulfilling all of those key steps of viral replication that we talked about in the last video lecture. But then if you're a herpes virus, you've got some little interesting twists to how you accomplish those steps. All right, an example negative strand RNA virus. Let's talk a little bit about influenza viruses briefly. Remember we talked about the negative strand RNA viruses. So if you're a positive strand or a plus strand RNA virus, when your genome gets inside the host cell is going to see that as a messenger RNA, and so it can immediately be translated to make the proteins of the virus. If you're a negative strand RNA virus, that's not the case. So you come into the host cell, that negative strand RNA has to actually be replicated. You have to make the plus strand or positive strand complementary copy, and then those the cell will treat as messenger RNAs and make the proteins off of them. All right, so what are some unique things about influenza virus, or interesting things about influenza virus? Influenza viruses, flu viruses, do have negative strand RNA genomes, but they're also segmented. So we haven't talked about that before. So this is supposed to be an influenza virus particle over here. It is an enveloped virus, and it has more than one kind of glycoprotein sticking out of its surface. And on the inside, there's a capsid in here, and packaged up inside here we have, these 8 pieces of RNA are packaged in capsids. And there are 8 pieces of RNA in here. And those 8 pieces of RNA will have separate genes on them. Okay, and so what this means is all 8 of those pieces of RNA go inside the host cell. They have to be converted over to plus strand RNAs, and then ribosomes will translate, make the influenza virus proteins from those. All right, and because they're negative strand RNA viruses, in order to make more of these guys, these RNA genome segments to package up into new flu viruses, they have to go through the minus strand RNA to plus strand RNA to new minus strand RNAs, like we talked about before. Remember we also talked about how if you're an RNA virus and you're making new complementary RNAs from an RNA template by reading an RNA template, that's something that these kinds of viruses do that our cells don't do. And so they have their own special RNA polymerase enzymes that do that. And lucky for us, because those aren't enzymes that our cells need or use, if we can come up with drugs that will interfere with the virus's RNA polymerases and not ours and our host cells, those might be good targets for drugs that would help work against the influenza. We do have some influenza drugs that are out there, as you guys know, but we need more. There's a lot of intense research going into coming up with safe, effective drugs for things like the flu. All right, another thing I want to point out that's interesting about influenza. So we hear all the time about how the flu changes every year. So there are a couple of major reasons for that. One is as these RNA genome pieces are being replicated, so they're going from minus strand RNAs to plus strand complementary copies, and then those get read to make new minus strand RNA genomes. The RNA polymerase enzymes that do that are very error prone. So influenza virus genome segments undergo pretty frequent mutations. Now sometimes those mutations just screw the virus up, and so any new influenza virus that has incorporated these genomes that have those mutations in them that are harmful to the virus, they're not going to be viable. They're not really going to be capable maybe of causing influenza anymore. And so just by natural selection, they're going to go away. They're no longer going to be useful forms of the influenza virus. But sometimes those mutations are beneficial. Okay, and so remember the genes on these RNA genomes encode things like these spike proteins that are poking out from the envelope of the influenza virus. So, and your immune system, the way that your immune system detects influenza is by identifying these spike proteins. So if influenza mutates a gene that's coding for one or both of these spikes on its surface, then your immune system may not be able to recognize those spikes anymore. So that can be advantageous to the flu virus. It's a way that it is able to escape the immune systems of its hosts. So that's one of the reasons why when you get a vaccine for flu one year to help prevent you from getting the flu, the next year that vaccine may not work so well because during the course of that year, these influenza viruses have been infecting human hosts and mutating so that the immune system weaponry and so forth that developed in people who had the flu that year will no longer be very useful if people get exposed to the newer strains of the flu the next year. All right, and so then another major way that flu can change. Now, you guys heard about the H1N1 flu epidemic, of course, a few years ago. And because influenza's genome is divided up into multiple pieces, every once in a while, there is a respiratory tract cell somewhere where you can get two very different strains of flu that infect a single cell at the same time. They go in there. And so flu number one, its little RNA genome pieces are replicating. Flu number two, its little RNA genome pieces are replicating. And when you start assembling the new flu viruses in that cell, you may get RNA piece 1, 2, 3, 4, and 5 from virus A and 6, 7, and 8 from virus B, or any other possible combination. So you get this shuffling of these RNA segments around, and every once in a while out pops a very different strain of the flu that has a different combination of these genes. And these new strains, sometimes they may cause more very severe types of flu, or it may be that people have had little to no exposure to these strains of flu in the past, so very few people have any kind of existing immunity to these new flu strains. So when you hear about a radically different strain of flu that has emerged and almost nobody has any kind of immunity to it because they've never been exposed to any kind of flu like it before, a lot of times that's what's happened. And the H1N1 flu we heard about a few years ago so much, that's been traced. What was the origin of that? And the last thing I read, it came from the index case, what we call the index case. The first person who had that illness was a farmer in Mexico who was simultaneously at the same time infected, had a cell in his respiratory tract infected with a human flu virus strain and then another flu virus that was a combined pig flu and bird flu strain. You guys have no doubt heard about the bird flu too. That's been in the news recently infecting chickens in the Midwest, at least at the time I'm making this video clip in 2015. So anyway, you had two flu strains, one human, a human flu strain, one a combination of bird and swine flu. They got together in the same cell in this farmer in Mexico, or maybe it was in started swapping these RNA pieces with each other, and out popped this new strain of flu that the world had not been exposed to in a very, very, very long time. And so then it started circulating around the world from there. Or I may have part of the story wrong there. It may have been a pig that actually was the original host for those two strains of flu, and they got together and had their little flu gene swapping going on, and out popped this new strain of flu that then infected the farmer. Anyway, I can't remember all of the details of the story. But anyway, that's a way. Every once in a while something like that happens, and out pops a much more severe strain of the flu. Okay, then how about HIV, human immunodeficiency virus? Let's talk a little bit more about it. This is an example of a retrovirus. So if you remember, in an earlier clip we mentioned that retroviruses have RNA genomes, but when they enter their host cells, those RNA genomes are converted into DNA. So they do the reverse of transcription by going from RNA to DNA. So an HIV virion, as depicted here, has two copies of its plus strand RNA genome inside a capsid, this little structure here that kind of looks like a thimble. It's an enveloped virus. It has glycoprotein spikes sticking out of the surface. One of those is called GP120, and that very strongly attaches to a molecule called CD4 that are present on some cells in our body. Primarily, these molecules are located on a type of T cell or T lymphocyte, which we'll learn more about when we cover the immune system. So if cells in your body don't have CD4 on the surface, they're not going to become infected with HIV because there's nothing for HIV to stick to. HIV also has something called a co-receptor, so there's another molecule on the surface that HIV also attaches to, and it has, some strains prefer to attach to a co-receptor called CCR5. Some strains prefer to attach to a co-receptor called CXCR4. We're not going to get into the details on that, but there is another molecule that HIV also uses to help it attach to the host cell and begin to enter the host cell. All right, so once that happens, you can see you have the fusion of the envelope with the membrane. Out comes the capsid. The RNA genomes of the virus come out. Here we see reverse transcriptase being mentioned. That's an enzyme that converts those RNA molecules to double-stranded DNA, depicted there in yellow. And then HIV makes an enzyme called integrase, which will integrate. So we now have a DNA copy of the genome. Integrase basically functions like molecular scissors. It cuts the host cell's DNA, and that viral DNA gets inserted into a chromosome of the host somewhere. So it sticks itself in your own DNA, and then the host cell just starts treating that HIV genome that is inserted into its own DNA like some of its own DNA. So this is a way that HIV is hijacking these normal processes. All right, so the HIV DNA that is inserted itself into the host undergoes transcription. So we have new viral RNA molecules that come out, and those can be translated to make new viral proteins, like the capsid proteins and those GP120 spikes that stick out on the surface. And then you've got to package up new HIV virus particles, just like we've seen before, and those get released from the host cell by budding. That's kind of in a nutshell how HIV carries out its life cycle. Reverse transcriptase, all right, that's the enzyme that converts the HIV RNA genomes over to DNA. And again, that's something that our cells don't use for their normal functions. This is something that HIV has that its host cells do not. So reverse transcriptase has been a great target for anti-HIV drugs to combat HIV. Many of the drugs that AIDS patients take work by interfering with reverse transcriptase. because if this enzyme gets knocked out, then HIV cannot complete its life cycle. And unfortunately, you may have heard before that HIV patients, when they go on these, there are a number of different drugs out there for HIV patients that target different things. But unfortunately, this reverse transcriptase is very error prone. So when it is replicating when it's doing reverse transcription, it makes errors very frequently. So that allows HIV to mutate, and those mutations can allow HIV to become resistant to various drugs that we try to use to treat HIV infections. So HIV is really, really good at mutating. And another problem with it is, early in infection, you don't even know you have HIV, but it has gone through this integration process into the DNA of its host cells, and a lot of cells in the body before you even know that it's there or would be treated for it. And so those drugs that HIV patients take are good at preventing the creation of a lot of new copies of HIV, but we're not good yet at finding cells in the body where those HIV genomes are located. So you can take drugs if you have AIDS and beat down the amount of virus that's present in your blood and other body fluids with those drugs, but the genome of the virus is just kind of sitting there waiting, and if you go off of those drugs, it gets activated again, and you just start assembling lots of these new HIV particles all over again. So it's we don't really have a concrete cure yet for HIV, unfortunately, because of some of these features of its life cycle. All right, so those were just 3 example viruses, just to kind of give you a little bit more of a feel for the diversity in terms of how viruses accomplish their replication. And just keep in mind, all viruses have to enter the host cell. They're going to have to go through those 5 major steps we talked about in some way. They've all got to use the host cell's ribosomes to make their proteins. Viruses don't have protein production machinery. In some cases, they'll also use other DNA replication and transcription machinery and things like that as well. That's going to vary from virus to virus. How do viruses differ? So all these different things we've talked about, host cell receptors, the genome types that they have, the strategies that they use for replicating those genomes, like we just mentioned with HIV and influenza virus, very different from what we saw back in Chapter 8 when we were talking about replication of the DNA genomes of living things. So lots and lots of variation out there. A couple of other little things that I wanted to point out, and these are discussed in your textbook in more detail. There are some viruses that cause cancer. You've probably heard before about how human papilloma viruses, or HPV, can cause cervical cancer in women, and that's the basis for the Gardasil vaccine that is supposed to help prevent infection with the strains of human papillomavirus that are most likely to cause cervical cancer, and so that's a good thing. But there are other viruses that can cause cancer as well. There's a virus called Epstein-Barr virus, or EBV, that in some cases can cause a disease called B-cell lymphoma, where B lymphocytes, a type of white blood cell, become cancerous and divide in an out of control manner in the body. So there are a few cases of this, of viruses that can cause cancer. And the ones that do that generally have genes that are sometimes called oncogenes. Onco refers to cancer, like an oncologist would be a cancer doctor. So these are genes that are involved with the trait of a cell becoming cancerous. They're called oncogenes. And generally these code for proteins that promote uncontrolled cell growth in some way. And I'm going to kind of leave that. We're not going to delve too much into how viruses cause cancer. You'll read a little bit more about that in your textbook, but we're going to kind of leave it at that. Another concept from your reading, cytopathic effects that viruses have on their host cells. So we've been talking about how viruses go into their host cells and hijack them for replication, and you might gather that wouldn't make a lot of cells too happy. And I know we also talked about how some viruses exit from their host cell through lysis, so that's definitely going to make their host cells unhappy if the host cells wind up dying and bursting as part of that process. But that would be considered a type of cytopathic effect. Cell-pathic, like pathogenesis or pathogen. It's like cell disease, cell sickness effects of viruses infecting their host cells. So different viruses will cause different cytopathic effects. This is a picture from your textbook, which is showing you that some viruses will actually cause host cells to fuse together. And that can happen because if you think about it, like when a virus is budding out of its host cell, it might attach to another nearby host cell that is really close, and then the membranes of all of those things fuse together. And so there are some enveloped viruses that cause the host cells to fuse, and those big giant fuse cells are sometimes called syncytia. Some cause their host cells to round up and look real grainy if you were looking at them under the microscope. So lots of variations. Anything that you could observe changes in the host cells caused by a viral infection can be called cytopathic effects. Now, some viruses actually aren't all that horrible to their host cells. You know, for example, hepatitis B virus infects cells in the liver, and it's fairly friendly to its host cells. The cytopathic effects are not all that severe. But as you guys know, hepatitis B virus is a very serious illness. And as it turns out, your immune system responding to hepatitis B virus is what really causes most of the damage, because your immune system is programmed, as we'll see toward the end of the semester, to torpedo cells in your body where viruses are located, to go ahead and sacrifice those host cells that have already been taken over by a virus to try to help prevent spread of the virus to other cells. All right, so that's going to conclude that video lecture clip. I know that one was a little bit longer than I like to have them go. But in the next video clip for Chapter 5, we're going to talk a little bit about how you work with and cultivate viruses in the laboratory. It's very different from what you do for things like bacteria and fungi in the laboratory. More expensive, complex techniques have to be used.